In the previous exercise, we have constructed the matrix table for all the data that's required for our study. In this exercise here, what I'm going to show you is how to normalize the raw count into a Z-score, which will allow us to compare across all the different cell lines. For the normalization, we need two specific software. One of it is the R program version 2.9.0. Another one is the shRNAseq algorithm. Now, it is important that the R program version 2.9.0 is required. Any other later version might not work because the shRNAseq was written using the R2.9.0 version of R program. So you can download this software in the link that is provided here and install into the program. So once you have installed, then I will show you how you can actually format it, the data set and to perform the normalization. Okay, so to find the R 2.9.0, you can go to this link here. So which will lead you to the page that look like this, basically. Now, so the current version, okay, the current version is actually quite advanced. I think it's the R 3.5.0. So, but what we need is basically we want to go to the older version, which you can click here. So you can click by this link here, which will lead you to the archive. So the version that we want is 2.9.0. So you can go down here, the list, and then when 2.9.0, which will be here. And then when you click this link, then it will automatically download into your download folder. All you need to do is that you extract it and then you can install it into um, your computer. And you can also similarly, okay, download the algorithm for the shRNA-seq using this link here. So once it is downloaded, so your file would actually located in the C drive under the program X S6, and then you can go to the R folder here. So now you will see the folder with R 2.9.0, and then if you go in there and you go into the library folder, that is where you would be able to find the shRNA seq library. So once you have downloaded and installed all the software that is required, you can refer to the Word document, which I have already uploaded to the Dropbox. The name of the file is called Lentiplex Analysis 2018-0430. Here you will get a more specific instruction on how to run the analysis and also the command for the R. So of note is that while this is the R program, it is slightly different uh, than the R suite which we use in the previous exercise. So what we need to do here is basically we need to process, okay, the information or the data from the matrix table in a specific format so that the shRNA-seq can actually read it. So if you want to access how all those how the format can be done, you can go in here. So under your program files, okay, R.2.9.0 and then library, and then under the shRNA-seq, there will be a folder on data. So under the data, you have, okay, one is the shRNA library. If you open this up, this is basically the file that will tell you, well, the annotation that you need to put in for your shRNA. I will show you how to construct that later on. And you also have a file which is called phenol data. Then you have two files, okay, which label as MCF7 viability time 1 and 2. So we will stick to the same format, okay, by changing just the data inside it to avoid any complication. So in the next few slides and the video, I'm going to show you how you are going to construct each of these files for your analysis. Okay, so to construct the input file, there's a few things that we need to put in mind. Okay, the exact protocol was actually written in this Word document. Now, first of all, there are the four files that we need to construct. One of it is the shRNA library, and within the shRNA library, you must have the columns on reference, pool, target seq, symbol, and also the gene description. Now, for phenol data, okay, we will remain as it is. We won't do much of the changes, and also for MCF7 wild time one, okay, we will also use back the same file name because all the script, the R script that is written was actually referred to specific file name. So in that case, we will try to change as little or probably stick to whatever file name and also the output that is, so to minimize the changes in terms of the script. So to show you how we can do it is basically the first thing is that, well, for certain reason the shRNA-seq would not be able to recognize the number under the so-called reference column. So to do that, what I'm going to do is I need to create a few more column and also rename a few. So first of all, the first column shRNA ID, I will change it to reference basically. So okay, so I'm just going to rename the column, and then the other one I will, so okay, the pool. Now I'm going to insert another column here, okay, called pool, okay, and then the target sequence, okay, symbol, and also gene description, respectively. So here you go. So that is what we will create a column. Now under pool here, we will all assume that it is just from one pool. Oops, sorry. So okay, so all copy all the way down. So this will be all assumed as a single pool. All right, then we can delete this one here. Now, as I mentioned it before, okay, the reference we need to create is more of a text. So what I'm going to do is I'm going to add at the front of the number with a few alphabets. So in this case here, I will use the concatenate formula. So by adding TRC in front, followed by the number. Here you go. So you can see that now I'm going to change it to here like this, and then I'm going to copy this whole column and then paste it as text over here. Okay, so I can delete this column. Okay, so you see that while I have now modified the matrix table slightly different to suit for the shRNA library seq input file. Okay, before we move on to the next stage to create the input file, note that some of the gene symbol have a comma with a different alternative name as well. Now, well, to avoid some of the problem in the downstream, so basically what we are going to do is we are going to create only one input for each of the gene name by removing the rest of the alternative gene name. So what we are going to do is we are going to copy the whole column here, and then we are going to separate them by, okay, text to column. We are going to separate them by comma. Okay, so now here you go. So it's all separated, and then we will copy this file back to the original analysis page, and then paste it as Word as text. So now that you have a matrix that is more suitable for the input into the shRNA-seq. Okay, now to create the input file, what I'm going to do is I'm going to copy, okay, all this column, and I'm going to create for BXPC3, for example, the input file. I'm going to paste it into a separate sheet, so I can remove this top column here, and then now uh, the next thing what I need to do is basically I need to remove those with the zero count because the shRNA-seq would return an error when it encounter a zero count here. So basically from both the plasmid and also the sample count, whichever column that have the zero, I need to remove them together. Basically these are all the all the row with the zero count. So I'm going to delete the row. Okay. So, and again, I'm going to do the same as well for the sample count here. So to remove all those with the 0. Okay, if you do not remove the 0 count entry, so the shRNA-seq will not be able to proceed basically. Okay, so now that is all deleted basically. Now, okay, I'm going to copy the first few columns, okay, and then paste it into the shRNA library file here, which is a text format. Okay. So now it is important is that this shRNA library uh, annotation must match with the input that I'm going to put in for both the control and also the treated cells. So I'm going to save this. Now I'm going to create another few more columns. So I'm going to create one column here. This is reference. And then again, so for each of the input file, you must have a column for reference, rep1 and also rep2. So I'm going to create a rep1 and then rep2 here. Okay, so the control, so this is the data from the control. Since the data set only have there's no replication, I will put okay, I will input the same value into the rep2 as well. So these three columns would then represent the control or the plasmid sample count, and then I will create another few more row. Okay, now this one will be for the sample count. So again, I will copy and paste it here. Okay, so for the control, I'm going to copy the whole data set and then paste it into the MCF7 wipe time 1 here. Basically, just going to copy and paste it here. Okay, and save. And then for time 2, so I'm going to copy and paste the sample count here. Okay. Okay. So now, okay, with that, so we did not touch the note that we did not touch the phenol data file here, so we will leave it as it is. So the input file should be ready to proceed with the shRNA-seq algorithm now. Okay, so now that all the input file is ready, we will open the R program and the script. Now, to get the exact script again, you can refer back to the Word documents here. So if you do not change all the file names and anything, so all you can do is that you can actually copy and paste the whole script and it will run it automatically. So, okay, now let's see if we copy the whole script here, and then I'm going to put it, paste it into the R here, basically. Now then it will take a while, so basically it will run it script by script, okay, each line by line. Okay, so we will wait for it to process. Okay, we'll show you some graph here. Now, for the detail of how to interpret the graph that they shown here, you can actually refer to this paper, okay, which is actually by Sim et al. In Genome Biology. This was a paper that was described, okay, how the shRNA-seq work and so forth, the algorithm, and what does it mean in terms of the data and the graph. So I will leave you to, it is good that you read through it and understand the principle behind it. But to cut short of our discussion, basically, if you see here is that while the report is actually automatically reported into the C drive, okay, users and whatever users name that you log on to and under the documents. So you can actually here, basically you can what we call it, well, you can actually retrieve the report from the My Documents folder here, and when you open it up, so and this is the normalized data that you can copy this and then you can paste it into an Excel file if you want to. Now, but if you do that, you will get a lot of the data which look like this. So all you need to do is basically just text to column and then convert, use the tab delimiters to actually generate into each of the columns. Okay. So now that you can see that, okay, so this is the reference, okay, this is the target sequence gene symbol. This is the original data that you input it in, and then there is a few columns in terms of normalization. And the most important thing that we will be looking at is actually this column here called the score. Now, if you refer back to the Sims et al. Paper, the score is basically referred to the C-score, and we will actually use this C-score, okay, for the subsequent analysis, okay, with RSA and also rigor. You can repeat this whole process, okay, with each of the cell lines, and then once we are done, then we can compile the C-score for each cell lines, okay, and then we can perform further tests from there onwards. That's it for now.